So welcome to the Department of Laboratory Medicine and Pathology Grand Rounds. DLMP Grand Rounds is a multi-site shared venue that promotes discussion of basic science and translates studies applicable to laboratory medicine and pathology and other departmental research and educational initiatives. We are excited to have Dr. John Mills join us and provide us his perspective and his thoughts on the evolving field of autoimmune neurology. And just some background on Dr. Mills, he received his Ph.D. in biochemistry and chemical biology from McGill University in Canada. And then he completed 2 fellowships here at Mayo, including our clinical chemistry fellowship and the clinical molecular genetics fellowship. He then joined the faculty and is currently co-director of the neuroimmunology laboratory, where he focuses on the development of assays to aid in the diagnosis of autoimmune neurologic diseases and related disease mimics. And a significant focus of his work is dedicated to identifying clinically relevant biomarkers and developing new ways of detecting them and then translating those discoveries to novel clinical assays. And so as a result of his efforts, his work has led to several first-of-their-kind clinical tests offered only through Mayo Clinic laboratories. And then in addition to his role as co-director of the Neuroimmunology Lab, he supports genetic testing for neuropathy and neuromuscular disorders in the Molecular Technologies Lab. And so with that, thank you again, Dr. Mills, for speaking today, and I will turn it over to you.
Thank you, Ellie, for the kind introduction. So the title of my talk is Setting the Pace in the Rapidly Evolving Field of Autoimmune Neurology. Much of my talk relates to work that's been done in Mayo Clinic's Neuroimmunology Lab, which is located on Hilton 7 in the downtown campus. The lab was founded in the 1980s by Dr. Van de Lennen, and it's really played a central and critical role in driving forward the field of autoimmune neurology since its original founding, and that continues to be the case today. So what this has triggered is really, over time, pretty substantial increases in testing volume, increases in the number of different testing platforms, increased testing complexity within the lab, and a constant influx of novel laboratory developed tests. And I just want to point out that none of this would be possible without our exceptionally dedicated and resilient staff. One of the benefits of being our lab is we always have cool, new, exciting things. One of the hard things is we always seem to have a lot of work because we're growing and we're always adding new things. So just a shout out to the lab. All right, so sorry. I have one financial disclosure related to some licensed IP from the binding site, but that's not related to the talk today. And I will be discussing briefly some uses of immunotherapies. So the learning objectives for today are really to describe participants should be able to describe the evolving field of autoimmune neurology, explain why identifying neural antibodies is clinically important, and justify the need for in-house lab-developed tests for the detection of these antibodies. So I'm going to start off just explaining what autoimmune neurology is. So autoimmune neurological diseases are those that impact the nervous system. So that includes a variety of different components of the nervous system. That can include the central nervous system, the spinal cord, as well as the peripheral nervous system. And then kind of the end of that is at the synapses where the nerves innervate the muscle at the neuromuscular junction, and can also include the muscle itself. So this kind of is an all-encompassing field of autoimmune neurology. And then within each of these subsets of the nervous system, you can have different presentations that can include, for instance, in the CNS, in the spinal cord, encephalitis, seizures, movement disorders, myelopathies, autoimmune dementias. In the peripheral nervous system, the autonomic nervous system, this can include axonal and demyelinating neuropathies, gastrointestinal dysmotility. And then when you get down to the muscle and neuromuscular junction, that can include myasthenia gravis, Lambert-Eaton myasthenic syndrome, immune-mediated necrotizing myopathy. So a whole variety of different clinical presentations, and they're not always one or the other. You can have multifocal presentations where you could have involvement of both the neuromuscular junction as well as the central nervous system. So they're relatively complex diseases. What triggers the autoimmune neurological disease itself is an aberrant immune system directed against the nervous system. Typically, this is antigen specific and driven. There are often triggers, aberrant immune responses. They can be paraneoplastic, so they can be associated with an underlying cancer that elicits a strong immune response that bleeds into the other parts of the body and the nervous system. Or it can be idiopathic, where we don't necessarily know what the ultimate trigger for the disease was. The identification of antibody biomarkers is pretty key in deciding on how to treat these patients, prognosticate for outcomes, as well as directing a cancer search for those antibodies that are associated with underlying cancers. So identifying an antibody biomarker is really central to the field of autoimmune neurology. Interestingly, though, only a fraction of suspected autoimmune neurological conditions do we actually have an antibody biomarker for clinical use at this point. But there are many more that are waiting to be discovered. So I'll get into this a little more, but we have evidence that there's many other antigen targets that we have yet to discover and develop clinical assays, but that's one of the goals of the laboratory moving forward. These conditions are important to identify because they are often treatable and manageable, especially if detected early. And that can be a big differentiator between a non-treatable permanent disorder or a neurodegenerative disorder where the patient's course can't readily be reversible. So it's very important to find these. So I'm going to start off with a couple cases just to illustrate different examples of what patients with an autoimmune neurological disease can present like. So the first case is a 38-year-old man. He started noticing fullness in his ears and hearing changes during the summer. By winter, he started having frequent frontal headaches and a gait imbalance. By springtime, he was noticing double vision when he tried to look left or right. We saw a neurologist. An initial MRI of the brain was performed, but it was unremarkable. They also did a CSF analysis, which identified 10 oligoclonal bands, as well as a mildly elevated protein, which is indicative of an inflammatory, potentially autoimmune condition. Other laboratory testing didn't provide insight into the specific disease etiology. The other thing to be noted for this particular patient is they also noted that the patient had seminoma. So this is just some of the pathology findings, just demonstrating the presence of a burned out testicular cancer. So this person basically had a previous history of testicular cancer. And then the second case I'm going to present was a 34-year-old man who presented with tingling and numbness of the feet. Several months later, this progressed throughout his leg, and he began to have notable weakness. He had mild tremors in his hands. He was dysarthric, so difficulty speaking, swallowing. He had electrophysiology studies that showed delayed or prolonged distal latencies, as well as reduced conduction velocities following some electrophysiological studies. The CSF analysis of the patient showed a relatively highly elevated protein concentration of 348 milligrams per deciliter. It's normal white blood cell counts and glucose levels. This patient was suspected of having an autoimmune disease right off the bat and was treated with intravenous immunoglobulins and oral steroids, but did not respond well to those. MRI of the lumbosacral nerve roots and the plexus demonstrated an abnormal finding. Now, these images aren't from this patient, but this patient did present with a very similar presentation. So on the right side is a normal MRI of this region. You can kind of see these thin, not particularly, not high intensity staining, whereas in this patient, you can see these are enlarged, and there's quite a bit of a intensity on the MRI, indicating that these are likely have become inflamed. This is indicative of an inflammatory process. So I'm using these two cases just to provide a little context that these disorders can be quite diverse, and really not so clear-cut clinically exactly what the patient might have. And that's where detecting neural antibodies is so essential in this field. In terms of who's likely to develop these autoimmune neurological conditions, it's not easy to predict. There are some risk factors, such as personal history or a family history of an unrelated autoimmune disease. Certain cancers are known to be more likely to trigger neural autoimmunity, so such as small cell lung cancer. Smoking can be a risk factor, certain HLA alleles, and more recently, the use of immune checkpoint inhibitors. So these are certainly not definitive, but they are pieces of information that a clinician will incorporate into their clinical exam to really try to pinpoint what the underlying disease etiology is for a patient. There's other things that can help out. So in terms of when to expect these or suspect these, if there's a subacute onset, that is also often a telltale sign. That's not always the case. And then there are specific neurological syndromes or presentations I've listed below. I'm not going to go into detail, but some of these are often indicative that the patient may have an underlying autoimmune neurological condition, as well as a fluctuating course or some of those risk factors that I mentioned before. So when a physician does suspect that the patient does have an autoimmune neurological condition, some of the things that they might immediately look into performing to help rule out or rule in the disease would be imaging. That could include imaging of the brain, could be the spinal cord, could be the muscle, electrophysiological studies such as electromyography, nerve conduction studies, or electroencephalograms to look at seizure activity. If there is suspicion potentially of a cancer, they may do a PET or a CT scan to look for cancer. In addition, CSF studies are used to identify inflammatory markers such as elevated white blood cell counts or oligoclonal bands, as well as elevated protein concentrations. Cancer screening, often patients that do have autoimmune neurological conditions also do have an underlying cancer. So ruling those out and identifying those is critical. And then potentially an immunotherapy trial where an initial immunotherapy might be given before it's understood entirely what the underlying cause of the disease would be. Neural antibody testing plays a central role. And in order to accomplish this, you really do need to test both serum and CSF. That offers the optimal sensitivity and specificity. And one of the benefits of neural antibody testing is when we do identify an antibody, they are typically very highly specific, and it can really help explain a very complex clinical presentation. So treatment depends on the disease, the underlying disease etiology, and identification of specific antibody biomarkers. But generally, this involves some form of immunotherapy, such as use of steroids, IVIG, plasmapheresis to eliminate circulating immunoglobulins, potentially B-cell depleting therapies, and others. If a cancer is found, not always the case, but generally removal of that cancer does often help with the autoimmune neurological. But it should be noted that responsiveness is highly variable, but can be to some extent predicted by the detection of specific antibody biomarkers. So in terms of disease mechanisms, So I mentioned earlier in the talk that often these can be parainfectious or paraneoplastic. So I'm going to just talk a little bit about those that are paraneoplastic in nature. So generally the theory on this, and this has been well proven, is that there's basically the paraneoplastic autoimmunity originates from a spontaneous anti-tumor immune response. And that immune response is initially directed against the cancer. And then often some of these cancers do express antigens that are highly enriched or expressed in the nervous system. And then you basically have a misdirected immune response against the nervous system. These are relatively rare. On the good side, though, they are associated with better cancer outcomes. So those patients that are mounting strong immune responses generally tend to do better. And then it's not all cancers that tend to be risk factors for these autoimmune neurological syndromes. It's enriched in certain types of cancers, and those cancers are often the ones that do express those onconeural antigens. So those antigens that are both expressed by the tumor that are also present in the nervous system. So some of those more common cancers associated with these diseases are small cell lung cancer, thymoma, breast cancer, gynecological cancers, and seminoma. I will mention seminoma particularly just because that is an immune, the testes are an immune privileged site. So generally, the immune system is not exposed to those antigens. So if you have a situation where those antigens escape and travel into the periphery, you can have that aggravated immune response. So the way that this is thought to work is basically you can have either B cell or T cell responses. And the T cell responses generally, again, reacting to these tumor antigens, you develop a cytotoxic T cell response. Those T cells then react to MHC1 presented peptides, and that leads to a cytotoxic T cell response that basically destroys the tissue that's expressing that epitope. Though the antibodies, biomarkers in these conditions are often directed against linear epitopes. And so we would use testing methodologies that are more likely to pick up those types of antibody responses. Those conditions generally are more difficult to treat. It's not the antibody biomarker is not directly playing a role in the disease. It's really mediated by the cytotoxic T cells. And on contrast, the B cell mediated responses generally result in circulating antibodies, and then those antibodies themselves are directly targeting the antigen that they're directed against, and may have a pathogenic role. And what I mean by a pathogenic role is that those antibodies could be acting as an antagonist of the target, an agonist that triggers an unnatural response, potentially can activate complement, leading to complement-mediated destruction, or antigen internalization, where basically the antibodies bind, they cross-link, and that triggers an endolysosomal degradation, but basically removes the antigen from the surface. Often these are surface or plasma membrane. In the case of these pathogenic antibodies, these are surface membrane proteins, often receptors that play key roles in cell signaling, and particularly in the nervous system. Those that are surface expressed targets that are mediated by the antibodies, where you have a pathogenic antibody, tend to be those that are more readily treatable. So this slide is demonstrating kind of all the commonly tested neural antibodies that we test in our lab, and presents a timeline of when they were discovered. So as you can see, when our lab was founded in the 90s by Dr. Van de Lenne, there was really a limited number of neural antibodies that had been discovered and documented to be associated with diseases. And most of them at that time were in green, those that target nuclear cytoplasmic proteins. So not antibodies that are directly playing a pathogenic role, but we're serving as biomarkers of a T cell mediated response. Over time, you can see a couple trends. Is one, there's been a huge increase in the last 15 years in the number of these neural antibodies that have been identified, as well as an increased number of those that have been identified that are actually pathogenic antibodies. So antibodies that are thought to directly mediate disease. They're targeted to the surface proteins where the antibodies can access those, bind, and influence the pathology. Not all of those antibodies are, though, associated with cancer. Only a subset are strongly associated with cancer. But generally, when you do detect those antibodies, there's a very high likelihood that that individual will have a particular type of cancer. I'm not going to go through all the antibody cancer associations. I just wanted to make you aware of that. So in the laboratory, the techniques that we utilize to detect these antibodies, we use a whole variety of different techniques. They're all shown here. I'm going to focus on two for today's talk, mainly indirect tissue immunofluorescence, where we detect the antibody binding a tissue composite. And when we see certain patterns of antibodies binding, that indicates what the antibody might be. And the other is cell-based assays, where we basically recombinantly express the protein on a cell, and we look whether or not antibodies bind those cells that are expressing that protein of interest. These other assays are currently used in our lab. They're common lab-based tests, but I'm not going to spend too much time on my talk discussing them. So tissue indirect immunofluorescence, the way that we perform this in our lab to detect these antibodies is we take a substrate. Generally, for our lab, the main method is to get frozen sections of mouse brain, put these on a slide. This is actually a composite of different critical brain tissues, as well as some non-brain tissues, to use as kind of a reference for neural specific staining. Those tissues then are then fixed and blocked, and then we incubate the patient's sera or CSF that contains antibodies. We apply it to these slides. We let binding occur. They're then washed off, and then we apply an anti-human IgG FITC conjugated secondary antibody that then detects any human antibodies that have bound that rodent substrate. And then on our microscope, we look at these sections and we look to determine if there are unique staining patterns identified that helps us determine what antibody specificity that patient might have. This is just an overview of a mouse brain. So I'm just highlighting some of the key areas that the lab uses to identify these antibodies. Cortex, hippocampus, the basal ganglia, thalamus, midbrain, and then the cerebellum as well. And these are all obviously physiologically key areas of the brain, but also common neural antibody target areas that help us distinguish between different types of antibodies. As an example of that, I'm showing here is a sagittal mouse brain section. This is not exactly the mechanism that we use in the lab to test, but I'm using this to illustrate really how we interpret these tissue immunofluorescence. So for a given antibody, we have a specific interpretive criteria. So in the case of AMPA receptor, we look for synapse rich regions of the hippocampus and dentate gyrus, shown here in 1, showing their staining. We would expect to find weaker staining, still fine staining, but weaker than hippocampus and the cerebellar cortex and the basal ganglia down here. And then we expect the thalamus to be negative. And then we expect to see specific staining in the molecular layer that's stronger than the granular layer. And if we were to see that pattern in the lab, that would be considered positive for AMPA receptor. And then we'd report that out and do confirmatory testing. This slide is just giving an example of a negative patient, as well as 5 different staining patterns for 5 key antibodies that we test for in the lab. And all I want you to appreciate here is that each of these has, when you look at the brain as a whole, each of these has a unique pattern. And that unique pattern really allows us to identify the specificity of the antibody. So tissue and direct immunofluorescence does have limited sensitivity for some antibodies, and we do have to use either alternative methods or adjunct testing to ensure we get robust clinical sensitivity and specificity. Some patterns are recognized, so the lab might get a patient sample, test it, look under the microscope, and identify a pattern that we've seen before, but we don't know what the target is yet. And those we don't report out. We consider those unclassified, and that we would then in the lab kind of determine if there is a particular clinical phenotype that fit with that staining pattern. And if there were, we try to develop confirmatory methods to define the antigen specificity of that pattern. And if that were the case, we then could potentially offer that as a clinical test. Some of the limitations of this approach is that for different antigens, there's probably not one optimized fixative. So not all antigens are created equal. And so there is the potential that we may not see something on tissue immunofluorescence just because of the fixation techniques that we use. Interpreting these is also confounded by coexisting antibodies or non-organ specific antibodies. And it really requires a lot of reader expertise, and that we do need confirmatory testing. So this is gonna bring us back to case one, which highlights some of the limitations and benefits of tissue immunofluorescence testing. So in this particular case, for this individual, they did order testing in our lab. They ordered an encephalopathy panel that includes a comprehensive battery of neural antibody tests that are associated with encephalitis. And when the lab tested this, what they found is no reportable antibodies, but they identified a neural tissue pattern that the lab had seen previously and determined it sparkles. And over time, this kind of sparkles pattern, where you see these punctate staining in particular regions of the brain, such as the periventricular parenchyma, the perihippocampal regions, and the region of the corpus callosum, where these kind of sparkly patterns were enriched. What the lab noticed over time is that patients that showed this typically were males. They often had hearing loss. They often had rhomboencephalitis that involved the cerebellum as well as the brainstem. And some of these patients had documented cases of seminoma and testicular cancer, which all overlap with the presentation of case one. However, the antigen target wasn't known. And in fact, after there's many years of attempts trying to discover this, using a variety of different techniques in the lab that I'm not going to go over today, sorry. But we were never able to really identify the antigen, which again is pretty important for defining the specificity of the pattern and helping recognize it potentially a new autoimmune neurological condition. So using this approach, Drs. Pittock and Dubé, along with Tom Kreiser, one of our former developers in the lab, in collaboration with UCSF. UCSF had initially developed this technique. We had our lab had collaborated with them, and what they were able to identify is the presence of a novel neural antibody targeting this protein called Kelch-11, which is an intracellular protein. And they were able to identify this specific peptide that was enriched in multiple patients. And they then went on to confirm that this indeed was the case. So based on that, the lab led by Dr. Dubé and Dr. Pittock, the lab went back at all these cases and screened them and identified a cohort of patients that did have antibodies directed against Kelch-11 antigen. And consistent with the case that we presented, case one, most of these patients did have rhomboencephalitis. Some had limbic encephalitis. A large percentage had hearing loss. and a large majority had testicular cancer or evidence of having testicular cancer previously, with a high number having seminoma. A significant portion of these patients stabilized or improved with cancer. So this was really the first time the lab had been able to use the novel FIP-seq technique to identify a novel antigen. So it potentially had huge applications for us, as we are very interested in discovering novel antibodies. So I had mentioned that the lab had as part of the discovery of this Kelsch antibody, the lab had developed a cell-based assay, which basically, where you take an antigen of interest, you either derive it off of an iris or you fuse it to GFP, so you have a fluorescent tag. You transfect this into cells, so you have a mix of cells that express GFP along with the antigen, and you have some cells that don't express the antigen and they're non-GFP positive. You incubate these cells with antibody. In this case, we used fixed permeabilized cells. These antibodies then were washed off. Any antibodies that were bound, a conjugated anti-human secondary was added. And then under a microscope, these were visualized, and the lab would basically look at co-localization between the Kelsch 11 staining and the patient IgG staining. And if those co-localized, that was evidence that indeed the patient had an antibody directed against Kelsch 11. So having identified a novel antibody that was relatively common in terms of paraneoplastic neural antibodies, the lab anticipated this would be a high volume test. So we really needed a way to have an automatable assay that performed well. Some of the typical easy to automate assays the lab uses, such as ELISAs and tissue IFA, either lack sensitivity or robustness when we did some preliminary testing, and we needed some other method to detect these antibodies. There were not any commercial products available, and we didn't want to wait for an IVD to spend years to develop a test that could be used in the clinical lab. Also, there's often limited interest in IVD developing these tests because they're relatively rare diseases and a niche area of testing. And there was a definite clinical need to identify these patients as soon as possible, given the impact that having a test available could make on their prognosis and how quickly they were treated. So we devised an in-house high-volume cell-based assay using a stable cell line expressing the Kelsch GFP fusion protein. We plated these on an optical glass bottom plate, fixed the cells, incubated them with patient sera, washed them, added a secondary, again conjugated to a fluorophore, washed again, and then could analyze these 96 well plates. The reason we did it in 96 well plates is it could be fully automated in the laboratory if we went that way. And what we did is we used a novel in-house approach to do this testing. We took advantage of an instrument, an image express microconfocal instrument that had a variety of objectives, could analyze, could run 96 well plates, had the fluorescence lasers that were necessary for our testing, and then could do automated software analysis. And could read these plates in less than 5 minutes. So I'm just going to give you an example, real quick video of what this looks like. So basically, this is just reading through the 96 well plate. And what you'll notice for a given sample, for a given sample, if you look at the green, so this is the antigen expressing, where the antigen is expressed, and the red channel, which is the patient antibody. This is a positive patient where you can basically probably notice that this pattern of staining co-localizes, indicating a positive sample. So this is just an image output of those 96 wells. And what you can see is basically the GFP only are negative samples. And then the red yellow are where you have co-localization. So we can zoom in on those images and have a closer look. manually. And what you can see is, yes, indeed, patients with Kelch antibodies co-localized to this these Kelch expressing cells. And you can use this co-localization to identify positives. In the case of negatives, you still see those green foci of the antigen, but you don't see any patient staining. If you zoom in and look at merged images, so what you can see is these yellow kind of greenish dots indicate where the patient antibody is binding specifically and only to where the Kelch protein is expressed. And we devised an automated analysis where the instrument itself could identify these foci based on their size, intensity, and extent of overlap. And then we could calculate the number of co-localized foci to the total number of GFP foci. And we identified a ratio of 0.05, where wherever we see above a ratio of 0.05, it's highly consistent with the presence of Kelch antibodies, Kelch 11 antibodies. So this allowed the lab to provide a very robust high throughput testing platform. We were the first to market to develop this test using this automated microscope platform. This CBA is used as a screening test, and then we confirm using tissue immunofluorescence. For the lab, this is a pretty big step. It reduces manual reading, improves our testing turnaround time, and really is the first step into the automated CBA interpretation space. Not only that, the assay provided excellent clinical specificity. So now I'm going to jump back to case 2. If you remember, this is the patient with tingling and numbness of their feet, some tremor, weakness in their legs. They had nerve conduction studies that indicated some abnormalities, and the patient didn't respond to frontline immunotherapy. So this patient's presentation was certainly suspicious for an autoimmune mediated neuropathy. And one of the first questions that a clinician would ask is whether or not this is axonal or demyelinating in nature. Patients that, so nerve conduction studies then can be used. I'm showing a picture of one of our members of neuroimmunology, Dr. Chris Klein, doing some electrophysiological studies. And basically in patients with reduced conduction velocities and prolonged distal latencies indicates demyelination. So that really measures the intensity and amount or speed at which electrical signals travel across the nerves. Not only that, this patient had a chronic progression over 8 months, unlike our previous patient, which had more of a subacute presentation, and they had elevated CSF proteins. All of these facts were consistent with the patient having chronic inflammatory demyelinating polyradicular neuropathy, or CIDP, but with some clinically distinct features. And this is actually a patient of Dr. Divyanshu Dubey, who recognized that this patient had features that were consistent with neurofasciin 155 IgG4 mediated CIDP, or actually a nodoparanodopathy disorder. So our group basically, once we had a test available, actually went back and looked at a large cohort of patients and really did a deep clinical phenotypic dive into what these patients look like. Most had a sensory ataxia, so some gait issues, tremor, weakness in their either distally or proximally, male predominant, poor responses to IVIG. But they could be treated with B-cell depleting therapies such as rituximab. They typically had high CSF protein concentrations and the symmetrical thickening of their nerve roots, all which were present in case 2. Nerve biopsy features showed evidence of demyelination as well as axonal damage and infiltration of lymphocytes in the sciatic nerve. So just to kind of explain the pathophysiology a little bit better for these cases, the body of the nerve, the axon, and then the myelinated axon portion, and then the axon terminal, which connects the synapse. Electrical signals are transferred along this myelinated axon, and this is done pretty efficiently due to the myelination and the presence of nodes. These nodes basically are what enable transfer of that very quickly electrical signal along the axon. And this is just depicting the structure of that node, where basically you have attachment between the axon and myelinating glial cells in the peripheral nerve system. That would be Schwann cells that form this kind of this junction at the paranode. And this junction is critical for properly localizing voltage-gated channels, which allow efficient transfer of this electrical signal throughout the axon. So what happens in these paranode or nodal nodopathies is that this complex, this association between the glial cells and the axon gets disrupted by antibodies. So antibodies come in here, they bind and block this interaction, and that actually causes breakdown of the structure. This is an example of some electron microscopy from patients. Basically, I just want to highlight that in normal cases, you see these, it's hard to see, but there's these tight junctions. But in the case of neurofasciin IgG4 patients, you see that the breakdown of this junction, you see the space that shouldn't be there. You can see it up here as well. This actually mimics quite well rodent mouse models where neurofasciin has actually been knocked out. So again, in a normal mouse model, you have these tight junctions, and these neurofasciin knockout mice, you have disruption of those junctions. This is just some evidence that these neurofasciin 155 IgG4 antibodies are playing a directly pathogenic role. This group, what they did is they took human IgG4 from control patients and then IgG4 from neurofasciin 155 patients. They injected them into a rodent rat model and looked at the impact. And what they found, those that received the normal IgG4 were normal, but those that received neurofasciin 155 IgG4 had some gait disturbances. That really just indicated that these antibodies themselves were pathogenic. So diagnostic testing for neurofasciin IgG4 antibodies in early 2021, there was really a single clinical research lab in the US offering this testing. It was utilizing a Western blot approach. It's not really conducive to clinical labs due to its manual and subjective nature. And there are some specificity concerns using Western blot. There are no commercially validated kits or reagents available to test for these neurofasciin 155 IgG4 antibodies. And there's this critical need to develop a robust clinical test that has high specificity and sensitivity, help identify these patients and assure appropriate therapy. So our lab has previously developed live cell-based assays for detecting antibodies against conformationally intact epitopes. And these have proven to actually be recognized as gold standard assays for detecting some critical antibodies like MOG and aquaporin 4 antibodies. The benefits of these approaches is that they can be automated, both collection and analysis of the data. They're objective. They don't require somebody to look at them under a microscope. You can do these on live cells without needing to fix the cells. They're cost effective. And again, the most important thing is you can use a native antigen that maintains the conformation on the surface of cells. Disadvantage, these can only be done in reference labs and generally, or highly specialized labs, and the reagents have to be produced on site. So you have to have a cell culture facility. So the way this works, again, is similarly to what I explained before. Basically, you express your antigen. In this case, for neurofasciin 155, it goes to the cell surface. We use GFP as a fluorescent marker that the cell expresses the antigen, shown here. So this is the antigen, GFP. And then your control cells don't express GFP and they don't express the antigen. So what we do is we mix this population of cells expressing and cells not expressing the antigen. We put them in the 90 plates. We then incubate them with the cells alive with the patient's sera. We then wash off all the non-bound antibodies, come in with a secondary anti-human antibody conjugated to Alexaflor 647. And then we perform a basic flow cytometry analysis. and generate the raw data looks like here. This is easy to do in 96-well format. We can run a lot of samples at one time, and it allows us to use native conformation. So just to give you an example of what results look like using this testing platform. So here is a negative sample. So you have your GFP negative cells here and your GFP positive cells. So the GFP negative don't express neurofascin 155. The GFP positive do express neurofascin 155 on the surface. So they're separated out using the GFP measurement of GFP. So low intensity, high intensity. So these express our antigen. And on the y-axis is the intensity of the anti-IgG4 signal. So in a negative sample, you basically have low signal in the Alexa flora because you don't have IgG4 binding these cells. So both the cells with and without the antigen are negative. But in the case of a patient that has neurofascin specific antibodies that are of IgG4 type, basically will directly bind this population and cause an upward shift in the Alexa flora 647 signal, indicating the presence of an antibody that's selective to the cells expressing expressing neurofascin 155. So then what we do is we measure the mean fluorescence intensity of this population and compare it to this population, and we generate a ratio. So in a negative sample, the intensity of these two populations is nearly identical, so we get a ratio of around 1. And in a positive case, we can see upwards of 100 to 1,000-fold increase in signal. So we end up getting positive samples generally give ratios in the range of 100 to 1,000, and which is demonstrated here. So we have a cutoff of 2. So basically any sample greater than 2 is considered positive. So any sample where the signal of the GFP expressing neurofascin cells is twice that of the non-expressing are considered positive. So we developed a live cell-based assay in our lab. We showed exceptional clinical specificity. We've tested over 300 disease controls, all of them negative. We've now identified as part of clinical testing, as well as testing during development, greater than 30 cases that were positive on the assay, and all those cases had disease consistent with what's expected for CIDP or a notoparanodopathy. We went live with the test in this fall, and we're actually developing additional tests in this testing space. So I just want to wrap up talking about the patient outcomes and why the antibodies were so important. In case 1, we identified the patient had a diagnosis of Kelch 11 IgG rhomboencephalitis that was confirmed by a novel testing platform. The progression of the patient's disease was stopped with immunotherapy treatment. And it just highlights why early detection is critical for these T-cell mediated diseases where the damage can often be irreversible. In case 2, the patient had a neurofascin 155 IgG4 paraneoplastic that we confirmed by the detection of neurofascin IgG4 antibodies by a novel flow cytometry assay. This patient had their IVIG discontinued. They were treated with plasmapheresis and rituximab. And basically the patient's strength went back to normal on his most recent examination. And repeat MRI of the spine showed resolving nerve root enhancement and reduction in the size of those nerve roots and plexus. So the take-home message is the number of newly discovered antibodies continues to grow. Translating these discoveries into clinical tests is critical for optimal patient care. And in the field of autoimmune neurology, there really is limited commercial kits, reagents, and assays, and it's really up to the lab to develop these in-house lab developed tests so that we can provide these critical tests to our patients and pursue our mission. So with that, I'm going to wrap up. This is a photo of our group. It's somewhat outdated. We haven't been able to take an updated group photo due to COVID, but hopefully that will change in 2022. And I thank everyone for your time.
Thank you for participating. Please click the button below to complete the evaluation and obtain credit.